Recording in progress.
Well, good morning, everyone. We are going to continue with Sophia's Hill work, the master thesis of Sophia, which is understanding the role of top-down input in visual information process. So, Sophia, go ahead.
Okay. So, good morning. I'm Sophia, and I've done my master thesis about understanding the role of top-down input in visual information processing. I have structured the presentation into the following sections. First, I will give a brief introduction about the visual cortex, hierarchy, and the top-down input. Then I will present you the V1LM model, the visual cortex model, a bit. And next, I will move to explain some of the methodologies and the results that we obtained. And finally, I will give you discussion and brief conclusions. So visual cortex is the brain region responsible for processing visual information. It is located in the occipital lobe at the back of the brain. And at its most basic level, it can divide it into the striate cortex, or the primary visual cortex, and the extrastriate cortex, composed by a series of higher order visual areas. The pathway that information follows in the brain, that visual information starts at the eye. Light is focused in the retina. Photoreceptor cells convert this light into electrical signals, which are then transmitted to the lateral geniculate nucleus in the thalamus via the optic nerve. From there, the information is relayed to the primary visual cortex and the other higher order areas where information is processed. So these areas are different because they have different perceptive field sizes, or different orientation selectivity, prefer temporal and spatial frequency of stimulus, and also distinct retinotopic mappings, which means that different areas may be looking at different regions of visual space. However, all these areas are not equally different, but there are some studies that show that in mice, there is a hierarchical organization within the visual cortex. This is, in this figure, you can see experimental work where they gave an anatomical hierarchy score to each of these different regions. And this hierarchical organization allow us to distinguish between two types of information flow in the visual cortex. We can have feed-forward or bottom-up input that goes upwards in the anatomical hierarchy scale, and we can have feedback or top-down input that goes downwards in the anatomical hierarchy scale. And in this project, we will mainly focus on understanding the role of feedback or top-down input in the visual information processing. This is because it is known that top-down input is involved in a lot of processing mechanisms such as attention, contextual stimuli, or prediction. However, the specific role that it plays in each of these mechanisms is not fully understood. For this reason, the objective of our work will be to analyze a computational model of the primary visual cortex and the lateromedial area, which is one of these secondary areas. How are we going to do this? So we are designing simulations and measuring different visual effects. We are going to measure response latencies, the evolution of the network's firing rate, receptive field sizes, and size-tuning curves. And in addition, to specifically address which is the role that top-down input plays in this model, we are going to perform the simulations in the V1LM connected version of the model. And we also perform the simulations in a disconnected version where we cut inter-area connections to study the difference between these two models. In addition, we will compare our results with experimental results also from mice experiment, mainly from these two papers from Siegel and Keller. So now I move to explain a bit how the V1LM model works. But before that, I would like to say that the development and the training of the model is out of the scope of this thesis. It was mainly done by the Allen Institute and Javi, as he explained in the previous talk. But still, understanding how it works and how it was built is essential for being able to perform proper simulations and for interpreting the results. So the V1LM model is inspired in a primary visual cortex model developed by the Allen Institute, which we named the VILE model. This model was simulating the lateral geniculate nucleus, LGN, and the primary visual cortex as a cortical column containing over 200,000 neurons with 5 different cortical depths. So the extension of our model with respect to this one is that we incorporate a new area, the lateromedial area, as a cortical column with a smaller radius. And thanks to that, we can simulate top-down input in the model. So the types of neurons that we have in the cortical columns, we have 1 type of excitatory neurons and 3 types of inhibitory neurons. And with respect to the connectivity, we have recurrent connections within the V1 column, also recurrent connections within the LM column. And with respect to inter-area connections, we have feed-forward inputs from LGN to V1 and from V1 to LM, and feedback inputs from LM to V1. The units that compose our model, the units that compose the lateral geniculate nucleus, are not neuron models. They are spatiotemporally separable filters. And the units that compose the 2 cortical columns of V1 and LM are GLIF3 neurons, which state for generalized leaky integrated firing neurons with after spike currents. This is a very long name, but they are based on the most simple firing neuron model, which is the leaky integrated firing neuron model. This model simulates the membrane's voltage potential of a neuron when an external current is injected into the neuron. So the voltage of the membrane increases until it reaches a given threshold. And when this threshold is reached, the neuron emits a spike and the voltage is resetted. The new incorporations of this generalized model is that now we have this term that are the after spike currents. The after spike currents are defined by this dynamical equation, and each of them have a different time constant, which allow us to simulate currents at different time scales. With respect to the reset rules, it's very similar. When the voltage reaches a threshold, it is resetted, and the after spike current also have its own reset rule. The main advantage of this type of neurons over the typical leak integrated fire is that now we can simulate the slow subthreshold region of the membrane potential and the fast ionic currents that are produced after a spike has happened. So the connectivity of the model is all set and we have the structure. The next step is to train it. And what we train are synaptic weights. We train them to gray screen and through drifting gratings. And the parameters that we train are the recurrent weights within the V1 and the LM column, synaptic weights, also the inter-area weights between V1 and LM, and the synaptic weights that V1 and LM neurons have with noise sources. These noise sources are in the model because we need to account for all the brain that we are not modeling, but that in reality is giving inputs to the primary visual cortex or to the lateromedial area. The way that we train it is by minimizing a loss function with 4 different terms. So the terms, these terms indicate that we are fitting to real neural data from NeuroPixels data from the Allen Institute in mice. We are fitting firing rates distributions to spontaneous firing rates and evoked firing rates, so to the gray and to the gray screen and the drifting gratings. We are also fitting orientation and direction selectivity metrics of the neurons. And we also include a regularization term in order to prevent the voltage to explode and to keep the weights within realistic boundaries. So having said a bit about the model, now I move to explain the methodology and the results that we obtain. And I will start with the response latency. The response latency is the time that a neuron takes after a stimulus has been presented to emit its first spike. And we will do this measure to white and black flashes. So for this analysis, we will only take core neurons. The core is the inner part of each column. And we only take those neurons for analysis purposes because we want to avoid border effects of the column. In addition, the first step for doing this analysis is to select the preferred contrast of each neuron, meaning that we select that by computing if the neuron has the largest firing rate to the white or to the black flash. Once we know that, here you can see an overview of the methodology. So the stimulus starts with 1 second of gray string. And then a black or white flash is presented for 250 milliseconds. We perform 100 trials. And for each neuron, the way that you measure the response latency is by measuring the time to first spike after the stimulus has been presented. Since we have 100 trials for each neuron, we compute its response latency or time to first spike as the median over all the trials. And if in some trial the neuron does not spike in the stimulus region, that trial is discarded. Before doing further statistical properties, we need to we make a null model in which we ensure that the neurons that we select for the analysis are really responsive to the stimulus, because we could have time to first spike just because the neuron is having a large spontaneous activity, and we want to avoid that. So the results are presented here. Here you can see the distribution of the times to first spike of response latencies for each region. You have the results for B1, for LM, for LGN, and for B1 in the disconnected model. And here you can see the means, and the errors are the bootstrap 90% confidence interval errors. So we can compare these results with experimental results from Siegel's paper. And we see that at first sight, qualitatively, the order between regions is the same that we obtained in the model. However, it's not difficult to see that the absolute values are smaller in our case. This is mainly because our model starts at the thalamus with the lateral geniculate nucleus, but real experiments done with mice starts to count the visual information processing at the eye. So there are some regions that we are not accounting in this case. What we can really do and measure and compare is relative distance between different regions, which shouldn't be changed from the experimental results. And we do this, and we see that the difference between LGN and B1 and between B1 and LM lays within experimental errors of the delays that are obtained from experimental values. And we also perform a Welch test in order to address whether the difference between regions was significant or not. We saw that the difference between LGN and B1 and between B1 and LM was significant with a p-value that was smaller than 0.05. However, the difference between B1 in the connected and the disconnected model was not significant in this case. This means that for this specific visual effect, the role that top-down input plays is not making a difference, at least not a significant one. And this is mainly because, as we can see, the first spike of LM takes place later than the first spike of B1, so that neurons in LM don't really have the power of affecting, at least not significantly, the first spike of neurons in B1. With the same simulations that for the white and the black flashes, we can also compute how the network firing rate evolves after the stimulus has been presented. And we do it for the white flash and for the black flash. And there are many things that can be noticed in these simulations. For example, we see that LGN neurons react more prominent to the black flash than to the white flash, whereas B1 and LM neurons react with more strength to the white flash than to the black flash. Indeed, if we compute the percentages, we see that 33% of LGN neurons only prefer the white flash, whereas this percentage is 75 and 91% for B1 and LM neurons. In addition, we see that there is a strong synchronization for the white flash. This can be noticed here, and can also be seen in the raster plot of one of the trials. These 3 bars correspond to these 3 peaks that we see here. This is something that, at least with this strength, is not biologically realistic. So it might be that it's a model flaw that we might want to fix in the future. And for fixing it, it's good to know the origin or the cause of this synchronization. And we can see that this synchronization is more pronounced in the disconnected model than in the connected model, when the synchronization seems to decay a bit more, meaning that probably the origin of this synchronization is not in the inter-area connection dynamics, but rather on the recurrent connections within the B1 column. We also notice that neurons react faster to the black flash than to the white flash. You can see that the peaks here happen after 50 milliseconds, and here happened before. And this is something that has also been observed experimentally, which I think that is a very important result, because you need to take into account that the model was not trained with flashes, only drifting ratings and gray screen. And it has still implicitly learned that it has to react faster to the dark flash than to the light flash, as it happens in biology. Now I move to explain a receptive field analysis that we did. And from this analysis, the measure metric that we are interested in is the area of the receptive field of the different regions. So a typical stimuli that is used for measuring receptive fields are Gabor patches, so which are circles with drifting ratings in size. This Gabor patch can be located in a grid of 13 per 11 different positions. And well, the methodology is as you can see here. This is visual space. This is what the network can observe. And the solid purple lines represent the projection of B1 and the core of B1 to visual space, and the dotted lines, the projection of the receptive field of B1 and the core of B1. So for each position, we will perform 50 trials of the stimulus. And for each position, and we will show the stimulus for each trial in 4 different orientations in order to avoid selectivity, orientation selectivity biases. So for each position and each neuron, we can obtain a pseudo raster plot like this one with trials and time. And we can compute an average firing rate over trials. This measure is telling us how responsive is the neuron to putting a stimulus at a specific place of visual space. So by computing this average over all the different positions, we can obtain a heat map or a matrix like this one that tell us how is that specific neuron perceiving the visual space. So this darker region means that the neuron is reacting stronger to a stimulus put in that region of visual space. And in this plot, the red dot is the projection of the neuron's position to visual space of that specific neuron. So the first step for measuring receptive field areas is to obtain a matrix like this one for all the neurons in the model. Here you can see 9 examples of this matrix that I just explained you. And as I said, this is the first step, obtain the original receptive field matrix. Then we have to apply a Gaussian filter to smooth the image. We are following a methodology that was as it was done experimentally. And we then binarize the matrix to obtain just zeros and ones by applying a threshold, that is the maximum receptive field minus the standard deviation of the smooth version. And finally, we compute the area as the size of the largest connected component. And this is an example for 9 neurons. In addition, we saw that there were some neurons that had inverse receptive field, meaning that they had a clusterized, well-defined region where they were actively being inhibited by a stimulus presented there. These neurons are also found in biology, and we needed to detect them in order to compute properly the area. Also, for this analysis, as we did before, we need to select only significant neurons with significant receptive fields. We again take only the core neurons to avoid border effects. We discard neurons with receptive fields larger than 2500 square degrees because are considered not to be biologically realistic. And we also apply a null model in order. I can give more details later if anyone wants to know about the null model. But the key thing is to know that this model will ensure us that we will only select neurons that have a receptive field with a clear structure and pattern in time and in space, over trials and in space. And that we will not take neurons whose area is just a product of having random activity because of spontaneous firing rates. Once we do this selection, here you can see the results of the receptive field areas. These are means and errors for each area, the V1 disconnected and V1 NLM, and here the distributions, and we can compare this again to experimental results. We see that the order is again the same that we obtain in the model. However, our receptive fields are much smaller than the ones that are obtained in reality. This is just because the original Ville model, from which we started to build our V1L model, was designed having smaller receptive fields than the biologically realistic ones. This at the point that was built, it was not the major concern to build a realistic receptive field. But now the Allen Institute is already working in a new B1 model where they are building realistic receptive fields. But at this point, it is still not fair to compare absolute values. But what we can do is, again, compare relative distance between regions. We see that in our model, the LM receptive field is 30% larger than in B1, whereas in experiments, this is 25%. And we also computed that in our model, the B1 receptive field in the connected version of the model is 7% larger than in the disconnected version, meaning that feedback projections, in this case, enlarge the receptive fields of B1 neurons. This is something that is not easy to compute experimentally, but we can do it in a computational model. In addition, when we see the results split in the cortical layers, we can notice that this difference between the disconnected and the connected model is larger in the deeper cortical areas, cortical layers. Okay, so now I move to the last part of the results and methodology, and I will explain, we computed size-tuning functions. Size-tuning functions are just curves that indicate how much does a neuron react to different stimulus sizes. We will use classical and inverse Gabor patch stimuli and of different sizes, as I said. And these different sizes will go from 5 to 50 degrees in steps of 5. Also, as we did in the previous analysis, we also need to select neurons that are only significant for this analysis. Again, we take only core neurons, and we take neurons only that we computed in the previous part that had a significant receptive field. In addition, in this case, we also restrict to neurons whose projection is centered in visual space. And this is basically because the stimuli is also centered in visual space. So now I will present you the results. Through looking to all the size-tuning curves that we found, we saw that there were 4 qualitatively different dynamics. And now I will show you an example for individual neurons of each of these 4 dynamics. In addition, for each neuron, you will see that there are 4 size-tuning curves. This is because we have a size-tuning curve for classical and inverse stimuli in the connected and disconnected version of the model. And this analysis was performed for B1 neurons only. So here, this is the first type of dynamics that we found. We see that the neuron increases its activity as the size of the Gabor patch increases, and it saturates approximately at the size of its receptive field, meaning that this neuron is not noticing what is happening outside its receptive field because it just remains constant approximately its activity after the Gabor patch has surpassed its receptive field size. For the inverse stimuli, this neuron decreases its activity as the proportion of gray screen that they see increases until it reaches also a saturation point, a minimum value that is close to the spontaneous firing rate of that neuron. The second type is this one, and it might seem similar to the first one, but there is a main difference, which is that we have a peak. And this peak is followed by a decrease of the firing rate. This is very important because it means that the neuron is indeed able to notice what is happening outside its receptive field in its surroundings. And the way that it notices it is by inhibiting itself. This is a well-studied phenomena in neuroscience and happens in biological experiments and is known as surround suppression effect. The third type are the size-tuning curves of neurons that had a receptive field inverse. We can notice the scale here that is negative, meaning that the activity of these neurons is always below baseline when we present a stimulus to the neuron. This is in agreement with the fact that we said that these neurons have an inverse receptive field, meaning that when stimulus is presented here, they are actively inhibiting themselves and having a background larger activity. The last type is similar. This size-tuning curves type is similar to the second one, but with the extra that now this neuron presents a surround suppression to the classical stimuli and also to the inverse stimuli. Here you can see the results average by neuron type. I put it the most relevant ones. So you can see, for example, that these two inhibitory neuron types have negative cystonin cores, meaning that the majority of neurons of these types have inverse receptive fields in the model. And we can also see surround suppression effect for some neuron types, especially in the connected version of the model and to the classical stimuli. This is very important, especially to notice that the surround suppression effect in all cases is more pronounced in the connected version of the model, which is the darker purple, than in the disconnected version of the model, which means that top-down input is the one providing this extra inhibition to the neurons so that they can react by inhibiting themselves to an stimulus in the surrounding of the receptive field. So in this case, we can say that for this effect, top-down input plays a significant role. Now I move to the discussion. I know I have explained lots of results, so I will go over the most important ones and discuss them. So for the response latency, we found that the values that we obtained of delays within regions were within experimental errors, and also that in this case, the top-down input did not play a significant role because the difference between the V1 in the connected and disconnected model was not significant. With respect to the firing rate evolution, we saw that there was a synchronization for the white flash, and that this synchronization was more pronounced in the disconnected version of the model. We also saw that neurons reacted faster to the black flash, which was an important result because this is also observed in experiments. And for receptive field and cystonin functions, we saw that LM mean receptive field is a 30% larger than V1, and this percentage is 25% in experiments. We also saw that LM feedback enlarged V1 receptive field in our model 7% on average, and that deeper cortical layers were more affected by this top-down input because the change of the receptive field areas was more significant in these cases. We also saw that the model is able to reproduce inverse receptive field with these two types of neurons as having the largest percentage of inverse receptive fields, and that the model is also able to reproduce surround suppression effect. And this surround suppression effect is more pronounced in the case of the connected model, meaning that LM feedback plays a role in this effect. Finally, some future improvements might be to fix the excessive synchronization of the model. By the time I did the project, this was still a problem, but the past few days, this has been solved by adding a new term in the loss function that penalizes excessive synchronization. We also found that there was an excessive dependence of V1 neurons on LM feedback. Here you can see the same neuron in the connected model and when we remove LM feedback. And what we can see here is that this neuron presents a blind spot here, where it doesn't even cover spontaneous activity. This is something that is not biologically realistic, and it's probably caused by the fact that we trained the model all together, so that the model is able to be realistic when we make simulations with all the model connected, but not when we cut some connections. So a possible solution for this might be to try a sequential training where we first train V1, and then we train the rest of the model. And as a long-term objective, it could be an option to increase the complexity of the model. Right now, our model looks like this, but the reality, as you can see, is way more complex in areas and also in connectivity within them. So as a conclusion, I would like to say that we analyzed the V1LM model by studying different visual effects. We found some similarities with experimental results, and we saw that top-down input played an important role for some of these effects. We also saw that the model has learned to reproduce some dynamics for which it has not been explicitly trained. And although some aspects need to be improved so that the model is still more biologically realistic, we obtain valuable information about the state of the model and the role that top-down input plays in its current architecture. And all the methodologies and the code that we develop, of course, will serve for testing future versions of the model. So as a final claim, I would like to say that I think that this thesis provides a groundwork for testing future versions of the model and doing a future analysis that bring this V1LM model to be the most biologically realistic visual cortex model. So, no, thank you.
Okay, thank you very much.